sick. So I just want to talk for a second about the next couple of weeks, what it's going to look like as far as what we're doing in here. So today we've got deciliation. We're going to knock the legs off these tetrahymenas. And then the following week we'll do feeding and vacuole formation. So what I want you guys to be aware of is that, so first the good news, no lab report for like 4 weeks or so. All these experiments are going to be built into one report. So it's going to be a real fat report, real nice. And you can of course work on that together with your lab partner. And then at the end of those weeks, we'll decide on a day. It'll be during normal lab time. We won't do lab, but instead you'll present on a little bit of some individual experiments that you design. And you'll take the most interesting parts just from your report, convert them to a PowerPoint slide, and then tell us about it, and hopefully you can make us interested. And Dr. Fungaday will probably be there to listen, but don't freak out. I have the say in the final grade, but she's going to be there. She may ask you questions and stuff like that. So I'm going to want to know the specifics of how you did your experiments when you design your own. We'll talk about that in a minute. She's going to want to know how you tie that back into genetics. So you kind of did the bioinformatics last time a little bit, and that has some information on the genetics of the organism, so you're going to want to tie it back into there. And I've also put in Canvas, it's a brand new module in here, so it's just free stuff. You don't have to read all this stuff. It's there to help you out. It's background information, genetic stuff you'll want to say. So let's talk about so for the next 2 weeks we'll learn the skills, like we'll deciliate this week and we'll feed next week. And so that's this stuff. We'll do growth in number, or mainly movement speed here this week. Next week we'll do feeding and vacuole formation. The 2 weeks after that, what I want you guys to do is per group come up with 4 experiments that you can do on the Tetrahymena, and then bring them to me. I'll help you hammer them out, make sure they're really nice and sciencey. So 2 weeks from today, we're going to do 2 of those. So what I want you to do is by next week have ideas to pitch to me so I can help talk you through, make sure it's really precise and scientific. And there are like 100,000 things you can test. So, and that's what some of this information is also for. So you can go in there and say, okay, how do I want to manipulate the organism to affect its growth number, or movement speed, vacuole formation, anything you can think of. Don't limit yourself in the ideas. Come up with everything you can, pitch all the ideas you can to me. And so hopefully by the end we'll have 12 unique experiments. They don't have to be, super genius, awesome, cool. I hope they are. They don't have to be. But they do have to show, some sort of results, some sort of experimental design, and some coolness in your ability to be a scientist and to design experiments. So it can be anything from you're going to make homemade media plates, or you're going to stain them with something, you're going to give them nutrients. It doesn't have to be complicated. You just have to be able to test a bunch of stuff. And we'll break that up into 2 weeks. We'll have 2 experiments a week. Next week, 2 experiments a week. And then one of the weeks I'll be out of town, so we'll either have a bioinformatics, again, the last pieces of the bioinformatics, and then after that present, or something along those lines. But I'll let you know more about that as we get closer to it. So that's the big picture. And then at the end of that, you'll have the big report and the presentation, and I'll try to get a more clear, like rubric, outline stuff I want to see in your presentation when we get closer to that. Are there any questions about that? I know it's brand new, it's probably unclear. Any questions about the experiments?
So today is both growth number and movement speed?
Today is mostly movement speed.
Okay.
Because there won't be really time to see how long it takes for them to grow. Mm-hmm. And so today will be movement speed, and the next week will be vacuole formation. Growth number is just something you can test. And you can do that by saying, okay, I'm going to make a slide of Tetrahymena. I'm going to look around. Okay, I see 20 running around. And then you come back again, you say, okay, now I see like 800 running around. So stuff like that, that would represent growth number.
That's just kind of be like over the week?
Yeah, that would be over the weeks.
Okay.
Or over days. They double really fast, and that's why we're going to try to keep them in the fridge, because they double every 2 hours.
Okay. Wow.
Which is crazy. Any more questions?
So each group has to come up with their own 4 experiments, or different than all the other groups?
Yes.
Okay.
And that's, like, there are tons of things in here that tell you things that affect them. You don't have to come up with your own. Like, there's stuff in here, I'll just give you random examples, because I printed out one of the things. Like, somewhere in here it talks about using a skimmed milk media. So you can figure out how to make that, or make a whole bunch of other medias. We have stuff you can add to them. Like I put on the thing, you could put glucose in some, you could put sucrose in some. There's iron chemicals that we have, and they like to grow with iron, and so we can put that in some. So it doesn't have to be huge tweaks, you know, in the experimental design.
Okay.
But yeah, so we won't start doing those until 2 weeks. So that's why you have all the time to come up with as many as you can, so that way we can make sure everyone has unique experiments and that they're scientific enough to be carried out.
Okay, cool.
Any more questions? It's going to be awesome. All right, I'm going to kill this recording.